Hello and welcome to the QIMR Berghofer Medical Research Institute High School Lecture Series 2020. My name is Professor Fabienne Mackay. I am the Director and CEO here at QIMR Berghofer. Unfortunately, because of COVID-19, we cannot welcome you to the Institute this year, but we are very pleased to deliver the lecture series online for you. In this series, some of our top scientists are going to talk to you about genetics. I'm not sure you're aware, but we actually inherit genes from our parents, and they come in sets, and they will determine who we are and why we're so different from one another. For example, they're going to influence the sorts of tastes and foods we like. They're going to influence whether or not we're going to catch colds or develop cancers. They're also going to influence how intelligent we are and the kind of personality we're going to have. Here at QIMR Berghofer, we are doing a huge range of research into genetics. Our goal is really to understand and discover which genes put us at higher risk of developing cancer, mental illnesses, and other conditions such as heart disease. In this series, you'll hear about our effort to use genetics to find new treatments for cancer, mental health problems, and how we are combining genetics and nanomedicines for new treatments. I hope we will be able to welcome you back to the Institute next year. But for now, be safe, and I hope you enjoy learning more about genetics. Thank you.
My name is John Pearson. I'm going to tell you a bit about butterflies and cancer. So my journey to science has been a little bit different. I started as a biochemist back before the internet, before mobile phones. I graduated, worked out I was rubbish at biochemistry, but I fell in love with this weird new thing that was just appearing called computers. So for the last 30 years, I've worked in science, but designing software for scientists to do data analysis, and that's becoming more and more important as we get more and more data. So let's talk a bit about butterflies and cancer. First, we'll start off with the human genome. So every single cell in your body has a copy of the human genome. There's about 30 trillion cells in your body, so that's 30 million, and every one of them has a copy of the genome, and it's identical. Every single cell has the same genome. The genome is made of a chemical called DNA. We represent DNA so we can work with it. We think of it as 4 letters, A, C's, G's, and T's. There's about 3 billion of these letters in the human genome. Remember, every one of your 30 trillion cells has a full copy of these 3 billion little letters. And in cancer, we get changes. So cancer is a disease of changed genomes, broken genomes, things that are different that make cancer cells behave badly. So let's do a little thought exercise to try and get a feel for that huge genome. If you think of 4 different coloured marbles, little marbles, just regular size. Let's go to the MCG, which is a huge cricket ground in Melbourne, one of the most famous cricket grounds in the world. It's about nearly 80 metres across, so it's a huge field. If you start in the middle of that, putting down marbles, 1 for each base in your 3 billion base genome, if you made this huge spiral, how much of the MCG do you reckon you'd cover? You'd cover 15 MCGs. So 15 of one of the biggest cricket grounds in the world, all covered in tiny marbles. That represents your genome, and every single one of your cells has a full copy of that genome. Now let's talk about butterflies. So genes are a bit like Goldilocks. You know the Goldilocks story? That porridge is too hot, that porridge is too cold, that porridge is just right. That bed is too long, that bed is too short, that bed is just right. Genes are just the same. Genes are like Goldilocks. They have to be just right. So this is a monarch butterfly, and this is a totally crazy example of how genes work. So on the left you have a caterpillar, squidgy body, 20 suckery feet, eats leaves. At some point in the caterpillar's life, it makes itself a chrysalis, sits inside there for 2 weeks or so, and what comes out is the butterfly. So it's almost like you've got two completely different animals. If you didn't know those two things, one made the other, you would never guess that. But there's still only one genome. So that squidgy caterpillar on the left has all the genes for making wings and having a hard body and 6 long legs and a rolled up squidgy thing that comes out to suck nectar out of flowers. All the genes for that are in the caterpillar, and at the same time the butterfly has all the genes for 20 suckery legs and squidgy bodies and eating leaves. The difference is genes get turned on and off. So you have to have just the right genes, express just the right amount at just the right time, and from one genome you could either get a caterpillar or get a butterfly. Now the lesson there for us is cancer is a bit the same. So to make our body work, we've only got one genome, remember, but every one of our different cells uses that genome a bit differently. The genes that turn on to make my lungs are really different to the genes that turn on to make my eyelids or grow my teeth or have muscles, but one genome making all these different cell types. So everything has to be just right. We need Goldilocks genes in order to get us operating normally. Cancer happens when that system breaks down. Either the wrong genes are turned on, or they're turned on too much or too little, not the right amount, or they're turned on at the wrong time, or they're broken. So cancer is a disease of broken genes. Because the genes are on or off at the wrong time or broken, cancer cells do things they shouldn't do. So we have accelerated genes. Cancer cells generally turn all those on. We have brake genes for growth. Cancer cells normally turn those off. Now those are normal functions. You want cells sometimes to divide like crazy and then stop. Think of if you fall over playing sport, you rub your knee, you end up with a big graze. Across time you get a scab, and underneath that scab your skin cells are dividing like crazy to cover up that gap. But then once they've covered up the gap, they stop. So that's good use of accelerator and brake genes. You want those skin cells all to turn their accelerator genes on and to grow like crazy to cover the injury. Once they've covered it, you want them all to stop. So that's normal behaviour, but cancer cells do it when they shouldn't be doing it. They avoid immune attack. Your immune system is really good at killing badly behaving cells or killing bacteria or viruses. Cancer, the genes that help cells evade attack by the immune system, cancer turns all of those on. They divide forever. Some cells in your body, you want them dividing. So blood turns over, so you're always making new blood and old blood cells are getting recycled, so that's great. You want them dividing, but you don't want a lot of other cells dividing like crazy. You don't want extra teeth. Once your cells have made the number of teeth you want them finished. Okay, so there's a real limit to how many divisions you want your cells to undergo. They become invasive and move around the body. Again, you want some cells to move. So your immune cells, you want them to be able to move around the body and rush to wherever there's an infection. But you don't want most things to move. You don't want to wake up in the morning and find one of your eyes has moved and is sitting on one of your fingers. So most cells in your body, they don't move at all. Once they've got their place, they sit and it's over. They grow new blood cells. This seems crazy, but cancer cells actually make themselves new blood cells. Once they form a cancer, they make new blood cells so they can get fed. So making new blood cells, that's, making new blood vessels, that's a good behaviour in some cases, but it's not a good behaviour for cancer, because you'd rather cancer died, but cancer actually convinces your body to make it new blood vessels to get it more food and oxygen. And cancer escapes from checks to make misbehaving cells die. So your cells have really complicated mechanisms of checks and balances. They're continually watching to see that the cell is behaving correctly, and if a cell is behaving incorrectly, it should be programmed to die, or its neighbours will tell it, we don't like the look of you anymore, you're not behaving, we think we should go. But cancer evades all of that. So cancer cells ignore all the signals that say, if you're a bad cell, you should die. Okay, so why do you need computer scientists? So we know now a bit about cancer, bad genes, broken genes. Why would you need a computer scientist? So this is old school. This is encyclopedias, books. So one encyclopedia, a big, fat, hefty, 500 double-paged book, is about 8 megabytes of information. to put that in a more modern context, if you took all of the sort of just under 3 million articles in Wikipedia, in the English language Wikipedia at least, you'd say that's about 17 gigabytes, or about a little over 2,000 of those huge encyclopedias. When we're doing a genome experiment, which is looking at cancer and trying to find all the broken genes in one person, it's 300 gigabytes. So it's 13 copies of the English language Wikipedia, and that's to look at one person. Okay, 37,000 of those encyclopedias. So once you get to that sort of amount of data, no biologist can look through that. That's when you start needing computer scientists. So we now have this nexus where biology and computer scientists work together, and that's what my team does. I work very closely. My team is all computer scientists, but I have a colleague and her team is all biologists, and for the last 10 years, the two groups have worked together. So we have biology and computer science working together to answer the same questions. If I want to actually do a study, one person doesn't make a great study. This is more what a study looks like. I now need 200 of those genomes. So it's nearly 750,000 big, fat, hefty encyclopedias, or about 60 terabytes of data. Okay, so butterflies are good, cancer is bad, but genes drive both. So when the right genes are turned on the right amount at the right time, we get butterflies. When the wrong genes are turned on the wrong amount at the wrong time, or they're broken, we get cancer. Thanks for listening.
Hi, so my name is Tracy, and I will be talking to you today about how I'm using genetics to try to find new cancer drugs. So we start with the cell, and each of us have around 3 trillion cells that make up our bodies, and in each of those cells we have a nucleus which houses our chromosome. And we have 23, not around, we have 23 chromosomes in each, pairs of chromosomes in each of our cells. And if we unravel these, then that is our DNA. And our DNA is our building block of life. It has all the instructions that make us who we are. And it's packaged very tightly. So in each of our cells, if we were to unravel the DNA, that would be 2 to 3 meters per cell. So you can imagine 3 trillion cells, 2 to 3 meters per cell. That's a whole lot of DNA that we've got inside of us. And so you can have sequences of DNA which can code for genes. And my research really focuses on the end point here, where we're looking at the actual base pair sequences, our DNA sequence. And if we were to type out this sequence into a book like this would be our instruction manual for how we were made. And if we put that, all of our DNA from one cell, all typed out, it would take an entire bookshelf. So if you think each cell has a whole bookshelf full of DNA in it. So I work on genetic variation, and this is single base pair differences between people. And if we think about our instruction manual, what we're looking for here are typos. So just one base pair differences between one person to the other. And these are inherited from your parents. So you inherit half of your DNA from your mother and half from your father. And so these are variants that you're born with. And there are around 10 million different variants in every person's genome. Most of them do nothing, so it's really normal to have variation between people. Otherwise, we would all look exactly the same, and we don't. So these variants affect different traits, so whether someone has different eye colour, how tall you are. It can affect things like whether you're a morning person or a night owl. Some of them do directly cause disease. These are quite rare. You don't find them in that many people in the population, but if you have one of these variants, it can directly cause a disease to occur, like cystic fibrosis, for example. Others just modify your risk, and those are the type that I'm interested in. So these are just whether you have a slight increase or decreased risk of developing a disease. So the disease that I work on is endometrial cancer. This is cancer of the lining of the uterus. It's the most commonly diagnosed gynecological cancer in the world, with around 380,000 women diagnosed each year. It's a relatively under-researched cancer compared to others like breast or prostate cancer, and there's only been 2 drugs approved for use for endometrial cancer in the last 30 years. So it's important for me that it's a disease that gets researched. And we research this through the Endometrial Cancer Association Consortium. So here at QIMR Berghofer, I run this consortium with 14 other studies from across Australia, Europe and the US. And it's through this consortium, what we do is, instead of all of these studies working individually across the world looking at endometrial cancer and trying to understand endometrial cancer, instead everybody comes together and we work as a team, and people share their data and share their patient information. And that means that we can make research breakthroughs a lot faster than if we were all working individually. So at the moment, we have just under 13,000 women with endometrial cancer, so some genotype data from them, we call them cases, and 100,000 women without cancer, and we call them controls. And what we do is test our genetic variants to see whether they increase or decrease risk of endometrial cancer development. And so if we have our DNA over here on the right-hand side, so at this particular point we have a C allele, and then in another individual at that point they have a T allele. And what we do when we test a genetic variant is we just look at that particular point in the DNA in all of our endometrial cancer cases, and also in our endometrial cancer controls, so the women that don't have cancer, and we count that up. And we can see that in the cases, most of the women there actually have a C at that point in their DNA. And compared to the T. And when we do a statistical test, we can say that yes, if you have a C at that point, then you are more likely to develop endometrial cancer. And it doesn't mean that you will, because if you look at the controls, we can see that there are about half of them that have that particular base pair there, and they don't have endometrial cancer. So it's not causing it, but it just means that you might be at a slightly elevated risk. So that's just one genetic variant out of all of the chromosomes that we have in each of our cells. So why look at just one? If we looked at them individually, it would take us a very long time, and we don't know how to choose which ones we would look at. So instead, we want to look at all of them, and we can do this using a genome-wide association study. And this is really cool. What we do is take DNA from someone, we put it into this machine, and it goes onto this chip, and we can look at millions of genetic variants all at the same time. So what we have is our one chromosome here, and what we can do is just walk along that chromosome and look at each of the genetic variants as we get to them, and then we plot out our results. And you can see on the y-axis here is our p-value, so this is a measurement that we get from our statistical tests. Each dot here represents a different genetic variant that we have assessed. And the red line here is what we're really interested in. We call this genome-wide significance. And dots that are above that genome-wide significance are things that we think are interesting and we want to look at in a little bit more detail. So that's chromosome 1. Let's look at all the rest of the chromosomes here. And so we've got this plot that looks a little bit like a skyscraper. So we call this a Manhattan plot. And what we're looking for here are the Empire State Buildings. We want to find the really tall buildings because they're the ones we think are really interesting. And when we find one, say this one here, we want to look at it in a little bit more detail. So there's our genetic variant that we've seen. We'll zoom in here. This is exactly the same plot as before, it's just been zoomed in. So each dot represents a genetic variant. On our y-axis, we have our p-value, and the red line is our genome-wide level of significance. And we can see there are a number of variants here that are above that red line. So we call this an association peak, because it looks a little bit like a mountain. And so we look at this area and we try to see what's happening here. Why do these genetic variants all seem to be associating with endometrial cancer risk? What genes do we find here? And there's a few of them here, so which one's important? And this is when we go into the lab and we do some experiments and we try to find out which gene looks like these genetic variants are affecting. And what we find is this gene over here, gene X, which is actually quite far away from these variants, but it looks like these genetic variants are actually affecting that gene. So what do we know about gene X? Well, we know that production of gene X is increased in endometrial tumour tissue compared to normal. And we also know from experiments from other people, other labs around the world, that if you inhibit gene X, so you block the activity of gene X, it will slow cancer cell growth. And they've seen this in prostate cancer and colorectal cancer, but nobody's looked in endometrial cancer. But we think that it's a pretty promising target, and so what we did was write to an American biotech company to help us develop inhibitors for this gene. And once we get these, we will test these on our endometrial cancer cells and see if it slows the growth of them. And so the ultimate goal is to try to use this information to develop drug therapies. So drug development is a very long process. It takes a very long time. So we're all the way down here at discovery, and we still have to get through preclinical, all these clinical phases, and then to the government regulatory paperwork. And that all takes a very long time. And it's important because you want to make sure that patients are getting safe and useful drugs at the end of it. It also costs billions of dollars per drug. At any point during this process, a drug can fail and it won't work, and then all that time and that money is effectively gone to waste. So what we want to start with are candidates that we think are more likely to make it through to the end. And what we know is that drug targets that are backed by genetics are twice as likely to be successful. And so that's why we're using genetics to choose what our candidates are at the very start of this process to hopefully make it down to the end successfully. And so that was just one of the genes that we're pursuing. There are another 2 that we found in a similar way, and we'll be investigating those in the same way. And this is not done just by me. I have a number of people that help me. So I have a couple of people in the lab, Dylan Glubb and Carly Chapman, and I also have 2 people that work on computers, because you can imagine with all of that DNA information that we have, we need people that are very skilled on computers to be able to analyse that. And we work under the mentorship of my lab head, Professor Amanda Spurtle. Thank you.
My name's Greg Anderson. I'm a group leader at QIMR Berghofer Medical Research Institute, and today I'm going to talk to you about nanomedicine. So you may or may not know what nanomedicine is, but you've probably heard of nanotechnology. And nanotechnology is where we're dealing with materials that are really very small. And to give you an idea of how small they are, I've shown some common objects here and given you their sizes in nanometers, from a cricket ball, which is about 100 million nanometers, right through to an iron atom, which is only a few tenths of a nanometer. And when we're talking about nanotechnology, we're talking about things in the 1 to 100 nanometer range. So the materials have at least one dimension that's less than 100 nanometers. And these materials have a wide range of applications in engineering, energy production, etc. Lots of biological applications and lots of applications in medicine as well. And that's why we have nanomedicine, a combination of nanotechnology and medicine. And there are lots of ways you can use nanotechnology in medicine. You can use them for sensing agents, as imaging agents, for drug delivery, and for tissue engineering. But we don't have time to talk about all those today, so I'm just going to give you a couple of examples of using nanoparticles to deliver drugs. But when we talk about nanoparticles, they're really very diverse. You can make them out of all sorts of materials, like organic materials like polymers and lipids, inorganic materials like silica or gold. Gold nanoparticles make really good imaging agents. But it's not just the composition you can change. You can change their shape, their size, their surface composition or surface structure, the charge on the surface. You can attach all sorts of things to the outside of them. So they're really very versatile, and the uses of nanoparticles are really only limited by your imagination. So what about some examples? The first example I'm going to give you is delivering an iron-removing drug to the brain. So why would we want to deliver an iron-removing drug to the brain? We know iron's important in your diet. If you get iron deficient, you can get anemic. So you need enough iron, but too much iron is also not very good for you. And we've known for a long time that increased iron is involved in the development of a whole range of brain disorders, neurological disorders, like Parkinson's disease. So we'd like to get rid of that iron. So how do we do that? Well, in this project, it was really a very simple way that we did that. We used a polymeric nanoparticle. So first we made our nanoparticle, a common polymer. You can buy it in a chemical company and mix it all together, just like cooking. sonicate it up and get a nice nanoparticle of about 100 nanometers in diameter. Then you load that with an iron-removing drug. It's called DFO. Sounds more like a shopping centre, but it's actually a really good iron-removing drug. It's been around for a long time. It's very safe. So now you've packaged the drug inside your nanoparticle. So how do you get this package to the brain, to the place where you need it to act? You need to put an address on it, and that's what these little blue things are. These are peptides, small parts of proteins that we've put on the outside of the nanoparticle to make it go to where we want it to go, to the parts of the brain. And in fact, these little peptides here come from a rabies virus, and they target the brain really well. Now we've got our nanoparticles, and we can test those out in a mouse model of Parkinson's disease, it's shown here. So here's a little cartoon just to show you what happens here. We take our nanoparticles, we inject them into the blood, they come along, they get to the brain, they can cross the cells lining the blood vessels in the brain, get into the brain and into the neurons, the main cells in the brain where the iron accumulates, and the drug gets released, it binds the iron, that changes its chemical composition, and it can diffuse out of the cells, out of the brain, and be excreted from the body. So it works really well. Before showing you how it works, I want to say something about targeting these nanoparticles to the brain. I could show you that in animals, but I'm going to show you this in a cell culture model. So here we're growing neurons, nerve cells, but in a big ball. We call them neurospheres. And these images here are these images here are cross sections through these neurospheres. And if we stain them with a blue fluorescent dye, you can see here, you can see lots and lots of cells. This dye stains the nuclei of the cells. And then we can take our nanoparticles and we can put a green fluorescent dye on those. So we have either nanoparticles without the targeting peptide on them, and those with the targeting peptide, and they're stained in green. And you can see the ones that have the targeting peptide, these neurospheres stain bright green, but there's very little green at all when it's untargeted. So the targeting makes a really big difference for these nanoparticles and can get them to the brain. And these nanoparticles do work really well. So here's some data with our Parkinson's disease mice. And we're not going to go through this in detail, but I just want to point out a few things. Firstly, on the top, we have brain iron. And let's just look at the orange bars here. You can see that our control animals, these animals that have not been treated with the nanoparticles, they have quite a lot of iron in their brain. But after treating them for a few weeks with our nanoparticles, which will remove the iron, that iron level has reduced quite a lot. It doesn't seem like a lot, but in the context of the animal, it's very important. In addition to reducing the amount of iron, we can also reduce the level of reactive oxygen species, and these are really reactive chemicals that you accumulate in the brain as a result of the extra iron that can cause damage to the brain. And does this actually make a difference to the behaviour of the animal? Well, it does. And here's one example of those sort of behavioural studies. In this study, it's called a rotorod study, we put the mouse on a rotating cylinder. And the mouse has pretty good balance, but eventually it will fall off. And we just work out how long, or time how long it takes for the mouse to fall off. And you can see here that here's, if we just look at the day 13 results, this is a completely normal mouse, not a Parkinson's mouse, and it can stay on this rotating cylinder for quite a long time, about 7 minutes. But here is a mouse with Parkinson's disease, and you can see it stays on the rod a much, much shorter time. So it really has a problem with its balance. But if we take those animals and we treat them with our nanoparticles that will remove the iron, the animal will stay on the rod as long as a normal animal. So we basically completely correct this behavioural problem with the animals. So these nanoparticles are really very effective at treating this particular condition. Right, so the second example I want to tell you is another, or talk to you about, is another nanoparticle study. But this time we called them DNA nanorobots. So it was a really cool name, and this study got lots of publicity. And in this particular study, we used these nanoparticles, these DNA nanorobots, to deliver the blood clotting reagent thrombin to tumours. And basically we're trying to kill the tumours. So what's happening there is if we can deliver this blood clotting agent specifically to the tumours, it can cause blood clots in the tumours. That stops the blood supply to the tumours, that they don't get their nutrients anymore, and they die. So it works really well. But this is much more complicated than the previous example that I showed you. And this took us 5 or 6 years with a big team of people to achieve this goal. So what did we do? And I'll briefly try and explain it here. We made a sheet out of DNA. So you've learned about DNA, you know you have a DNA double helix, and the two strands of the double helix use base pairs to hold them together. And we use the same principle to make this DNA sheet using lots and lots of pieces of DNA in a process called DNA origami. So we took this sheet, we attached some thrombin molecules to them, and that in itself is no good because if you put that into the blood, you'll just cause the blood to clot. So we need to protect the thrombin from the blood until it gets to the tumour site. So we rolled it up into a cylinder and we stitched it up so the thrombin could not be exposed to the blood, and then we used that to treat the tumours. So here's an example of what that DNA sheet looks like in more detail. I'm not going to go through this, but the different coloured lines represent different strands of DNA, and you can see there's some long ones and some short ones and different colours because there are different types of strands, but they all go together to make this really elaborate DNA sheet. Perhaps the best way to show you this is in a video. So here's our DNA sheet, and the purple things are the thrombin attached to it. In real life, we had 4 thrombin molecules. We curl this sheet up into a cylinder, we stitch up the ends. The green things on the ends were the targeting parts of the molecule that would allow it to lodge in the tumour blood vessels. We inject it into the bloodstream, it comes along, it gets to the tumour. That sheet then opens up, it exposes the thrombin, and that causes a blood clot to form. And this gets bigger and bigger and bigger, and eventually so big that it blocks the blood vessel. So that stops the blood supply to the tumour, and the tumour dies. And that's what's showing here. Here's our tumour dying. So it's a really neat way of cutting off the blood supply to the tumour. And there's our dead tumour. So we're scientists, we like numbers. So here's just a mouse model where we're growing some tumours and we're measuring the size of the tumour. And you can see down here that these are our DNA nanorobots in the black line. Tumour's still growing a little bit, but these are the controls that don't have the targeted DNA nanorobots, and you can see they're growing very quickly, but the ones with the nanorobots are growing very slowly. So it's really very effective. And if I just show you a picture of that, these are the tumours that have been cut out from the mice, and here's the controls. You can see the tumours are sort of big and pale, but after we treat them with the nanorobots, the tumours are really tiny and very dark because they're all shriveled up. So this was a really effective way of reducing the growth of the tumours in these animals. And it's a very complicated way of using nanotechnology, but it shows you the sorts of things you can do these days. So that's all I have time to tell you about today. The nanorobots may not quite be little machines that you might see in a science fiction movie, but they are still simple machines, and we're getting more and more sophisticated all the time with some of the things we can do with these really remarkable materials. So who knows, maybe sometime in the future you might be working in nanomedicine as well. Thank you very much.
Well, thank you. Hi, everyone. Welcome to this online presentation. My name is Eske Derks. I'm from the Netherlands originally. Today I'm group leader of the Translational Neurogenomics Laboratory. And I will explain a little bit more how we can use the genetic results from mental health studies to improve the treatment for patients with these disorders. So we've really made major breakthroughs in the last 10 years. So around 20 years ago, we were searching for the single gene linked to mental health disease, so the gene for schizophrenia or the gene for autism. But we have now learned it's much more complex, and we have moved on to use first candidate gene studies based on the biological mechanisms. But now we're doing genome-wide association studies where we measure millions of genetic markers and try to find links with mental health disease. We've also developed methods to estimate how much all genetic variants on a human's DNA contribute to mental health disease, and also to understand whether there's any genetic overlap between different mental health diseases, for example, between schizophrenia and bipolar disorder. So I really want to get a message across that there's a big difference between monogenic disorders, like for example Huntington's disease, and complex genetic disorders such as mental health diseases. And I try to illustrate that here by showing you two different towers. So as you can see, this tower here, which is made with Kepler blocks, you can imagine that if you take out one of these blocks, for example, especially at the bottom of the tower, and you take one out, that the tower will collapse. So you will have a disease. So one change is enough. Well, if you have this tower here, which is from a game Jenga, which you may know, this is much more complex. So you can push out these blocks and you can take away quite a few blocks and nothing will happen. The tower will still be there. So meaning that you can carry a lot of genetic risk factors while still not having a disease. So it's really different, like it's much more polygenic is what we say. So there's many genetic risk factors. So as I said, we are now using the genome-wide association study design where we measure millions of genetic markers. So you can see that here. So this is the DNA and there's millions of changes that we measure, and we refer to these as single nucleotide polymorphisms, meaning that's one mutation in one of the markers replaced by something else. And we measure millions of these. And then for each genetic marker, we will compare the frequency of the marker in a large group of patients with a specific disease and in a large group of controls, so people without a disease, and see if there's any differences in the frequency between these. We then make a plot to show the results for these millions of genetic markers, and we show the chromosomes. And here you see the strength of the evidence for an association with a specific genetic marker. So, for example, this genetic marker shows strong evidence of being different in patients compared to healthy people. So this is one example of a genome-wide association study. And this is a study I led a couple of years ago where we collected data on cannabis use. And especially in Australia, less so in the Netherlands where I come from, but especially in Australia, the media was very excited about the findings of this study. And quite funnily, it was referred to as a genome-wide association study in the media. So what we did, we collected information on cannabis use in 185,000 subjects and conducted a genome-wide association study to find the genes that are linked to cannabis use, whether you ever used cannabis in your lifetime. So the findings of the study, so we found 8 independent genetic markers that were linked to cannabis use, and there were 35 genes that were linked to lifetime cannabis use. So that's like whether you ever use cannabis during your lifetime, tried it even just once, or whether you never used cannabis. When you combine all genetic markers together, we found that these explain 11% of the variation in the population. So that's all markers together. And one of the most interesting findings for the genes was a link with CADM2. And that was quite interesting because this gene is highly expressed in the brain. We know that. And it has also been associated with other disorders, other substance use disorders. But also with a more risk-taking, optimistic, carefree personality. So it seems to be a gene that makes you try do different and new things. This is another example of a genome-wide association study, and this is for a disease that's a bit more serious than cannabis use. So this is bipolar disorder. And here you can see what we call a Manhattan plot that summarizes the evidence for all the millions of genetic markers, and everything above this line is significantly associated with bipolar disorder. So you can see that there's many genes coming out that are linked to bipolar disorder from this study. And that's really interesting. And also we can now start to ask the question, how can we use the results from this study, from this genetic study, to really improve the lives of patients with bipolar disorder or with another mental health disorder. Because at the end, that's what we want to do as researchers, not just find new things, but also use them to make the lives of patients better and to find better ways to treat them. So how can we do this? There's several different ways. So first of all, we can try to predict an individual risk by using genetic data. So in this example, which is on smoking and trying to stop with smoking, they used 5 genetic markers and combined them into a single genetic score. So people were put into different groups based on low, average, or a high genetic score. And you can see here that if you are in the high genetic risk group, that it's more likely when you smoke that you're also dependent on the nicotine, because not everyone who smokes will become dependent. But if you're in a high genetic risk group, you're more likely to become dependent. And then when you get treatment to help you stop smoking, when you are in the high genetic risk group, it will be more likely that you start smoking again after quitting. And also the chance of successful cessation will be lower when you're in the high genetic risk group. So you can see how we can use the results from genetic study to predict the individual risk. Another way is to predict treatment response for a specific medication. So this is an example on naltrexone that is used for the treatment of alcohol dependence. And when you take naltrexone, when you drink alcohol, you will feel really sick, so that helps you to stop drinking. But the effect of this medication will also depend on your DNA, so on your DNA profile. So, and there's a gene, it's a opioid receptor gene. The gene is called OPRM1. And when you carry a specific variant of this gene, which is called the G1, at least one G allele, you will have a lower risk of relapse when you use naltrexone. And when you do not carry this G allele, but you have different variants, genetic variants, you will have a higher risk of relapse again. And this is one of the few examples at the moment of a genetic test that is included in the clinical treatment guidelines. So in Canada, they will test you when you are prescribed naltrexone, they will test you for this gene, which variant you carry. So this is a field I'm very active in myself. How can we use the results of genetic studies for drug repositioning, for finding new treatments? And drug repositioning is also described as teaching old drugs new tricks. So that's one very famous example of drug repositioning. It wasn't based on genetics, but there was a medication, sildenafil, also better known as Viagra, and that was originally tested for hypertension. But when they prescribed it to patients, they saw in men that there was like a side effect. And you can probably think, know what kind of side effect that was. So after this observation, they started to use this drug, reposition it, for erectile dysfunction. So that's one interesting example of how you can use existing drugs to find medications for conditions that you cannot treat yet. So when you look at all these genes, and this is again the Manhattan plot for bipolar disorder, we have all these genes. So we can then look which drugs, which medications target the activity of these genes. And if there's any medications that target these genes, then they could be effective for bipolar disorder. So there's a library that we use, it's called CMAP, and they've done a lot of experiments in the lab where they had cell lines, like grew them in a dish, then added a medication and looked at the effect on the gene expression in this dish. And they shared that information, so we can now use that to link it with the genetic information on a disease. so that we can find better treatment. So the take-home messages, just the messages that I hope you will remember after this talk. So genetic risk factors play a very important role in mental health disease and also in substance use disorders. We know now that at least hundreds of genes play a role, it could be thousands of genes even, and the influence of each individual gene is quite small. There's now still a limited, but I think it will increase in the future, there's a limited number of examples where genetics testing can be used to predict whether someone will respond to treatment or not, and we refer to that as precision psychiatry. We can use the results from genetic studies to discover more effective treatment opportunities, and we can use drug repositioning to find existing drugs, and that will also help reduce the cost to develop new treatment, because for existing drugs we know they are safe. We already know they are safe for humans, so we don't have to go through all these expensive trials to establish safety. So thank you for your attention.